we're ready to go on to our next segment in the program, where we're going to learn the buzz on mosquitoes. Our next speaker is Anita Schiller. Anita is a dedicated biological control practitioner and Texas master naturalist who joined the Harris County Precinct 4's Park and Preserves team in the year 2000 and was chosen by Commissioner Cagle to lead the biological control initiative since its inception in 2013. Ecological systems with an emphasis on entomological vector interactions and the integration of biological control into vector management are Anita's focus. She received the Public Health Entomologist Specialty Certification in 2021 and serves Harris County constituents by developing biologic and environmentally friendly tools for mosquito reduction in precinct parks and preserves where conventional pesticide treatments are not the best answer. Her passion drives the program's research. Her focus on process design encourages insectary production efficiency, and she represents the program at industry, scientific conferences, and Wildscapes Workshop today. So please help me welcome Anita Schiller.
Good morning. This is going to be interesting. I should have brought a little podium. Well, it's not about me, so I guess you don't really need to see me. The question is, can you hear me all right? Yes. All right. I wanted to start off with a huge thank you for the invitation to allow me to share my spiel, my thing with y'all this morning. It has been a circular succession for me. I started out in the plant world eons ago, and ventured on into the entomological world. As Jaime mentioned in the introduction, vector interactions have been kind of my passion and have kind of just captured me over the years. But to be able to bring it all back into the plant world is just super exciting to me. So thank you for the invitation, and I hope I won't bore y'all too much. It sounds like we've got a bunch of plant aficionados in the room, but I hope that with this information that I'm going to share with you, I will bring you around into the bug world a little bit more and looking at a critter that we ordinarily cringe about when we hear the name. There's just so much more to that animal that we call mosquito with the cringe than we realize. And when I was charged by Commissioner Cagle over 10 years ago to look at biocontrols, I did the same thing as everybody else did, like, mosquitoes, really? Golly, what's about a mosquito? You smack it on your arm, it's a blood smear, done. You spray it with some Raid or some pesticide, you fog it a little bit, done. Like, what is there to know? And then I opened the door into the mosquito world, and, well, I don't think I'll ever come out of it. So the disclaimer for this program is that we know from an industry and a scientific standpoint an awful lot about the biology of mosquitoes, an awful lot about how to get rid of them, kill them. Pesticide industry is huge, the war on bugs and the war on mosquitoes, but we know hardly anything about its role as pollinator or other ecological roles that mosquitoes fill. We know a little bit, we have some glimpses, but the big drive for scientific inquiry really is on reducing the burden that mosquitoes put on human population. And so when I was presented with the invitation, I said, oh, this is amazing, I get to share everything I know. I was like, oh wait, no, I can't do that. That would be a book because there's a lot to know about mosquitoes. Excuse me. I'm going to try not to lose my voice. But bringing it back around to the plant world is really difficult because there's, like I mentioned, there's not all that much. So I hope you will forgive me that the plant portion is a small one. And I hope that maybe you all can help by bringing more information about mosquitoes as pollinators forward from this. So if you'd like, I'd go ahead and get started. All right, the first thing I want to start with is, again, the cringe when we say mosquitoes. lets us know that mosquitoes, not only are they a typical pest that we just don't like as a nuisance, but they do they do represent the greatest burden of animal kills to humanity globally. The chart is a little bit outdated as to 2018, I believe, and it's a lot more. It's over 1 million now. The global pandemic has kind of redirected some of our statistical analyses towards SARS-CoV-2 analyses. So over 1 million deaths globally are attributed to parasites and pathogens, human parasites and human pathogens that are vectored by mosquitoes every year. That is every single year. And some of those deaths are utterly preventable. Nope. But even though there's such a large burden, a public health burden produced by mosquitoes, there's only a handful of species that are responsible for causing those disease transmissions. It's less than 10% of all the species known, and we've got currently, taxonomically speaking, a little bit over 3,500 different species worldwide that have been described. And again, it's less than 10% of those that are listed and verified as competent vectors for disease. Although there's more, 243 potential vectors that we feel are part of the play in disease transmission, but they have not been verified as such. And there's a lot that goes into that. But the word that's highlighted there is competent vector, and we'll go into what that actually means. There is a specific description that goes with competent vector. Mosquito diseases and pathogens and arboviruses in our area are primarily those that are listed. Our area are the continental United States. Mosquitoes do not know political borders. They don't stop at the Texas border and say, oh, I can't go there. That's what we do, and that's what I'm currently faced with. Like, okay, I'm charged with Precinct 4. Really can't go into other Harris County precincts to do my work. Mosquitoes don't have that same challenge. What keeps them in check, of course, are ecological interactions where their habitat is no longer favorable. But malaria is our #1 imported mosquito-borne disease. So that shows that we're an area of travelers. Houston, Harris County is a great melting pot, and it's a city of commerce. So again, people traveling bring back diseases with them, unknowingly bringing them back. And so mosquito-borne parasite, malaria is the #1 reported disease. It is vectored by a very specific type of mosquito group, but the #1 locally transmitted mosquito-borne virus is West Nile virus. Raise your hand if you've heard of it. All right, okay. So the difference there between people bringing back malaria or getting infected locally means just that. When we're vacationing or in other countries, sometimes we let down our guard. We're not quite as vigilant. The same as we go outside and we're gardening, we're putting in those new Biden's Aristosas and Turk's cap we managed to buy at the Native Plant Society sale this afternoon. And we forget to put our insect repellent, or we don't like to use insect repellent for whatever reason. We actually do take a chance of getting infected with West Nile virus. That's Harris County specific. That's our main mosquito-borne disease that we need to worry about. West Nile virus arrived in 2013 and has made a beeline across the United States. This chart is from the CDC. It has not been updated for 2021, but it does show you the hot spots of areas. And you notice where, of course, Harris County is down here. We do have outbreaks of West Nile virus every 6 to 8 years or so, and those typically follow mild winters. 8 months after a mild winter is the predicted model that shows an uptick in West Nile virus transmission. And West Nile virus is a sylvatic a virus that hangs out in the forest, but it travels in the avian community. So birds move the virus around. There's a group of birds that's highly susceptible and another group of birds that are the amplifying host of the virus. But when they come through in early migration or during the summertime and congregate in watery areas, especially during drought times, is when West Nile virus transmission is the highest. And you can see that in here in 2018 map. Here we go. But not only can mosquitoes pass on these pathogens to us, a new study that just came out a couple months ago shows us that, well, gosh, humans but somehow manage to pollute the earth with plastics. Well, gosh, we'll just give it right back to them. You all heard of microplastics and nanoparticle plastics that are polluting our waterways? Well, mosquitoes are now known to be able to uptake those nanoparticles during their feeding behavior in the larval stage in the water. The microparticles transcend into the teneral adults, and they're able to give those right back during the next blood feeding. Isn't that something? Here we thought we're only drinking the microparticles in the water. Oh no, it's much more devious than that. But gosh, they've been around for a very long time. The historian and the audience will attest to that. Initially, we thought that mosquitoes followed the rise of mammals. So 45 million years ago, blood-feeding mosquitoes came into the landscape. But recently, and I believe this was found last year in this amber, we found an anopheline mosquito full of blood that goes back 100 million years ago. So y'all are old enough to remember this movie, Jurassic Park. You'll remember that premise on how he wanted to extract the DNA from the amber mosquito, although it wasn't a mosquito in the movie, and build his own dinosaurs, and all the scientists goes, oh, baloney! There weren't any mosquitoes around back then to bite dinosaurs. It's not even possible. Never mind all the other stuff. Oh gosh, maybe it was. All right, so here's the timetable, and it shows you where this brand new mosquito, not brand new, this very, very old one, but the last discovery, the 100 million year old discovery came into play. Again, it's an anopheline mosquito. And what that means, some of the terms that I throw out in the beginning, I will explain later on in the presentation. Incidentally, the ones highlighted right here in the red box are some of the most troublesome mosquitoes that we're dealing with nowadays, the most burdensome for humans. All right, alongside them coming off dinosaurs, following through the Ice Age, following mammals into the current era, they went along these paths of evolutionary preferences, of evolutionary adaptations that got some of the species to actually specialize in certain hosts to take their blood meal from. And we can see this in this phylogenetic tree, evolutionary phylogenetic tree, on how some of the blood preferences of mosquito species are relatable. We can form relationships on which mosquitoes bite which type of host and whether or not we can trace that back through their DNA. For instance, Anopheles scambii, you see it right here with the red line. It is known to be a preferred human host. It absolutely targets humans over any other blood host. Luckily for us, or fortunately for us, Anopheles scambii does not occur in Harris County. However, this guy right here does, Culex quinquefasciatus. It's listed here in a yellow line. You see its host preference are birds. So I made the marker here as birds. Culex quinquefasciatus, even though it has evolved to be taking bloods primarily from birds, is our primary vector for West Nile virus. Well, how does that happen? If it feeds on birds, why are we getting sick with West Nile virus? How odd. I'll explain that later. There's some other oddballs in there. Oh, I'm so sorry, I skipped. Aedes aegypti, for instance. Aedes aegypti, next to Anopheles gambii, is number 2 worst enemy that we have, globally speaking, when it comes to transmission of arboviruses, arthropod-borne viruses. Aedes aegypti, unfortunately, was introduced into Harris County, or into our area, during colonialization. So 150, 200 years-ish ago, not only did we bring a bunch of merchant ships, merchant marines, slaves from the old world, we also brought along Aedes aegypti in the ship vessels, in the potable water, drinking water vessels. We brought Aedes aegypti right along with it. And it came ashore along with the people and the goods and said, oh, I like it here. People are my friend because people are its preferred host meal. Oh, I'm so sorry, one more. Toxorhynchides amboinensis is classified here with the purple. It is a non-blood feeding. Well, why is that there? Where did that come along in the evolutionary scale? Non-blood feeding mosquitoes? How weird. I'll explain that later. So if you're, most people come to me and say, gosh, you know, you talk about mosquitoes being so scary. Mosquitoes don't bother me. And I'll be in a room of people in a party or something, we're talking, but mosquitoes don't bother me. And I go, and before I can say, oh, yes, they do, you just don't notice them, somebody else will interject and go, oh, no, they love me. They love me. They're always biting me. But why? Well, science has been looking into that, because if we can understand why something happens, then we can disrupt it, right? Then we can try to keep it from happening. So scientists have figured out or identified these 7 reasons why. First of all, mosquitoes, adult mosquitoes, navigate through their antennae. The neurons are in their antennae, their neuron receptors. So their noses are their antennae, as with most insects. And mosquitoes are really good at finding carbon dioxide. We mammals mostly exude carbon dioxide when we talk, when we sweat, and mostly in our breath. And mosquitoes are awfully good at that. There's some species that can find carbon dioxide up to 100 feet away. That's 30 yards. Golly. That's really far. There's no outrunning mosquitoes. Also, if you exercise or just exert yourself, the lactic acid that you get from hurting after you work out also gets exuded in your sweat. Have you ever been told by a fitness trainer, oh, if you're sore, if you had a good exercise, a good workout, you should work out again the next day to get rid of the lactic acid? Say it. Yes, everybody's worked out, right? Well, when you get rid of that lactic acid, the mosquitoes find you even faster, which please don't take that as an excuse for sitting down, right? Recently, we've learned that type O blood is highly attractive. Folks with type O blood are more attractive, doubly more attractive than the other blood types. And we're not really sure why that is. We just figured out that it is. It's a fact, but we don't know why yet. If you're already infected with an arbovirus of the flavivirus family, you become more attracted. If you're infected with Zika virus, chances are that the next Aedes aegypti and Aedes albopictus that finds you, that will be in the room, will go to you first, because it recognizes that you have that virus that it is programmed to distribute. How weird. And then, oddly, pregnant folks are more likely to get bitten than non-pregnant people. And they're not really sure what it is about the pregnant folks, what their biome is or their chemical cloud that makes them more attractive. We only know that they are. Mosquitoes find them faster. Now, here's my disclaimer. This is my single and only slide with flowers and plants. I'm sorry, I tried. However, this slide depicts the bizarre nature of mosquitoes. They are so interesting. So I mentioned that the antennae are their noses. They find their way around. They find us, or they find their blood meal by using olfactory cues that they find through their antennae. And that little bit has not escaped the plant kingdom. Up in the northern area, in the northern parts of our region, the United States, northern parts, the bog orchid uses the olfactory cues that the mosquito uses to its advantage. Where the bog orchids bloom, there may be a lot of other bugs, but they exploit the vast numbers of mosquitoes that come off the water at the same time. They exploit them. They make them their slaves by tricking them. They trick them by exuding through their floral scent nonanals on octanols, the stuff that also we exude in our sweaty feet, and the lactic acid compounds and the carbon dioxide compounds and all the other compounds, and ethanol compounds that we exude from our sweat and our breath. The bog orchid does that same through its flowers. So it's a mimicry and a trickery. Although the mosquito does get some nectar from it, it is tricked to pollinate the flowers. As it enters the flower, a little trigger mechanism will actually deposit a pollinia packet onto the forehead of the mosquito, like it hits them out of nowhere. And then it's got this dangly thing in front of it, and it goes, oh my God, get off of me, get off of me. And it goes and lands on the next place where it could possibly land, which happens to be the next flower, and then the pollination occurs. We have a cousin of this northern bog orchid here, Planetara flava, which acts similarly, has a similar pollination syndrome, although we have other pollinators and more pollinators as well in our area. I have been chasing down images of our Aedes communis population on southern rain orchids for, probably 15 years or so, and I cannot come up with a single picture. Can y'all tell me why? Well, the primary reason is the habitat is no longer suitable, and habitat fragmentation has made finding the southern rain orchid populations almost impossible. About 15 years ago, we knew there were about 15 small plot populations. In Harris County, I believe we're down to 3. Well, that's why. All right, so when the mosquito goes to probe the flower or take a blood meal, there's this huge process in play, and it has this enormous toolkit in front of its face. We call it the proboscis, which is not to be confused with the proboscis of a butterfly or a sponging fly. It's its own weapon of mass destruction, if you will. It has been superbly engineered and has 7 components, or 2 pairs each, of components that spread apart in a few seconds, spread apart the skin or the membrane through which it'll imbibe, or spread apart the mechanisms of the flower through which it will take in the nectar. But the components themselves are marvelous. If you've never seen a video, a close-up video of how a mosquito feeds, I highly recommend it. YouTube has several videos out there. But these things right here that look like they're all spread out really are a single tube or looking tube. It's a sheath that folds out of the way once it's done its job. It spreads apart. The mandibles insert themselves into the skin and spread apart. Then they're followed by the maxillae right here that are serrated with 47 teeth that go back and forth so rapidly. Yeah, 47 teeth on each side, not combined, on each side. So next time a mosquito lands on you, just watch it, how fast it actually penetrates. No, okay. Well, all that happens so fast, but then the very center is the business part. The thing that is pointed, has a pointed tip that then is able to finally penetrate the capillary, penetrate the blood vessel to extract the blood or the flower. Oddly, if you don't have a barrier or a membrane or a skin to push this thing out of the way, the mosquito cannot drink. How weird is that? I find it weird. I'm so sorry. I always say, how weird is that? And then people go, no, you. But anyway. That is my understanding, how people perceive me. So anyway, the mosquito either visits a flower or takes a blood meal, and then what happens? Here's the proboscis part. It sucks up the blood in this case, and somehow the internal mechanism of the mosquito actually understands whether it's a blood meal or a nectar meal or carbohydrate meal. The blood gets diverted into the midgut. The carbohydrate meal, full of fructose, sugars, and proteins, gets diverted into the diverticulum. What happens to the blood, though, is the really cool part. Again, I use cool because it's fascinating to me. It gets digested slowly, but within that blood, all kinds of things happen. There's a chemical process that separates all the particles. The red blood cells are separated out. The proteins are separated out. Furthermore, into the amino acids are separated out, and everything is compartmentalized. But the STDs, I'm so sorry, the viruses it took up, they do their own thing, but I'll get to that later. So you may say, well, all the mosquitoes bite, right? All the mosquitoes bite? All mosquitoes bite? Right. Of course not. Yeah, I set you up. You knew that already. Not all mosquitoes bite in order to lay eggs. There are some mosquitoes that are able to lay eggs as virgins. They have not had a blood meal yet. The ones that do take a blood meal really makes up the vast majority of mosquitoes in our that we know of. But the ones that can't come off the water to lay eggs without taking a blood meal, we call those facultative autogenous. The ones that do are autogenous, anautogenous species. That's a problem for vector control. So this thing right here, we have in our area, I say thing. It's Aedes atropulpus. We have them in our area. They're down in the salt marshes. They hang out in rock pools and crab hole areas. They can come off the water without taking a blood meal. However, when they come off, they could already be infected with a virus that has co-evolved alongside that species to transcend the oogenesis, that can transcend from the mother into the progeny. So it's a problem for us. We don't know where they are. When I say for us, for vector control specialists, we don't know where they are until they come off the water, and then we have to bop the problem up the head once they start biting people. Well, once they start biting people, it's kind of too late when it comes to intercepting arbovirus transmission. This guy, you're probably familiar with. Gosh, you're in Harris County. You probably make a annual trip down to Galveston to the beach. Have you ever been chased by black mosquitoes, hordes of black mosquitoes, and you run and you swat and you scream bloody murder, and yet they just keep chasing you, and when they do bite you, they hurt? That's them. That's them. Well, they're part of this group. Not only do they hurt when they bite, they haven't taken a blood meal yet, and they could already be incriminating in arbovirus transmission. Obligate autogenous group is the real bizarre, eccentric part. It's a very tiny fragment of mosquito species that can be lumped in 3 genera. Excuse me, and in our area, we only have one, and that's this guy right here. For those of you that have been around, that have known me, since I moved to Harris County 20-something years ago, know that I kind of dove into Toxorhynchites rudderless world, mosquito world, about 15 years ago or so, and this is the creature that I dream about, Toxorhynchites rudderless. It's fully autogenous. The cool part about it is, you see this proboscis thing right here? It's downward caved. Remember the previous slide with the maxillary, the 47 teeth on the side? It doesn't have that. It's more like a butterfly. It can actually slurp up floral nectar without having to destroy capillaries and skins along the way. This job right here, unfortunately, we don't have them in our area. They're in Southeast Asia. It is also fully autogenous, meaning it never takes a blood meal in order to lay eggs. See this club-shaped appendage at the end of its proboscis. It prevents it from taking a blood meal, and it doesn't need one. It's so highly evolved. It also gets its nutrients that are essential for vitellogenesis and oogenesis, egg development, during its larval stage. The interesting part on Malaya is they have evolved alongside chromatogaster ants that serve as ants and aphids in the rainforest canopy. Malaya will find the trees where chromatogasters are, chromatogaster ants. They will find the ants that are walking down the tree, fly up to the ants, harass them with their antennae, probe them with their club-shaped proboscis, and I don't know if they tickle them until the ants regurgitate or pee, or if the ant finally throws up its hand and says, I give up, here you go. They regurgitate the honeydew that they took up from the aphids up in the canopy, and Malaya feeds on that. Yeah, mosquitoes are pretty cool. All right, so I've thrown out a bunch of terms already. Maybe some of them you knew and some of them you didn't, and I'll explain some of that further on. But I thought it'd be a good time to actually figure out whether we know what a mosquito is. Raise your hand if you think you know what a mosquito is. Just play along. All right, pop quiz. We're gonna look and see if you actually do. You don't say it out loud, you keep it on your head. It's like watching a card trick. Just see the card that you think you want and keep it in your head and remember it. All right. These are the larval stages of flies. See if you know what a mosquito looks like. Oh, goodness, sorry, wrong one. Keep it in your head. Do you think you know? Who had it right? Everybody. Oh my God, this is so cool. So cool. Sometimes I throw these out when I'm at conferences, scientific conferences, in a room full of mosquito killer scientists. And there's this trend in science where, science got to be cool and sexy to be attractive to students or undergrads. And they get into genetics, DNA manipulation and all that is a cool, sexy thing, and which translates into that very few of them have actually ever seen a mosquito in real life. They don't leave the dang lab bench or the computer. And I throw that out there and people go, they look at these slides like, oh no. And I see them going, yeah, they have no idea of the animal that they're working with. But y'all do, so cool. How about adults? Everybody knows what an adult mosquito looks like. We've all killed them in our hand, right? In our arm. Keep it in your head. You got it? You got it? Who had it right? Yes? Oh. Let me guess. This one, right? Gotcha, right? The AKA the mosquito hawk, right? Who was taught that's a mosquito hawk and it eats mosquitoes? When I first came to Texas, that's what people told me. It wasn't until I really got into ecology and started joining master naturalist groups and really learning about the local area where I realized that all those old wives' tale, all that stuff that people tell you, and only half of it is true, and that one is probably one of the worst myths you could ever be told. That beautiful crane fly right there, mosquito hawk, it doesn't even have functioning mouth parts. How could it kill a mosquito? Not even in its larval stage. Gosh, it's really just a way of moms keeping their kids at bay. It's like, leave that thing alone. Don't kill it. Don't pull its legs off. Leave it alone. It's good for us. Gosh. All right, moving on. I'm going to run out of time, I can tell. All right, so now that some of us know or are validated that we know what mosquitoes look like, for the rest of those that don't, or are really quick in killing a mosquito before you get a chance to look at it, here's some of the main parts. Obviously, mosquitoes are insects. I'm just going to assume that you all know that part, right? Head, thorax, abdomen, wings, correct? Got it. However, what you may not know yet is that Mosquitoes belong to Diptera, their order of true flies, meaning they only have one set of wings, true wings, and an haltere, a vestigial wing on the end that helps it hover and navigate and steer in the air column. Also, mosquitoes, scales from top to bottom, backwards, forwards, head to toe, including their wings. Even the wing veins are scaled, and they have this beautiful fringe of scales on the hind margin of the wing. And sometimes the proboscis is even scaled, which can be used in identification. And disclaimer, I won't go into identification of mosquitoes in this presentation. We'd be here, through tomorrow. But that is the main part. All mosquitoes are full of scales. When we trap them in our traps, very often the scales are knocked off, and then we have to find other ways to identify them. And molecular identification of mosquitoes is now a thing. It's coming up for the last 5-6 years or so. We've been barcoding our trap kills because it's a little bit faster and more reliable than visual identification. But for taxonomy and for our area, I will reiterate, mosquitoes are true flies, or two-winged flies, and they all belong to Calicidae, and we have 3 subfamilies in our area, 3 subfamilies. The Anophelines with only 7 of them, and I have to say none of our Anopheles are human malaria vectors anymore. Okay, the Acacheson, Attacapans, and even Carancuas that were spoken of earlier avoided Harris County, downtown Harris County, like the plague in the summer. And I wish I had known that talk was happening. I could have really slanted this presentation a little bit more into the history of them. But they weren't in our area. Buffalo Bayou was avoided like the plague because the Anopheles that were in the area, Buffalo Bayou area back then, during the Native American area, still carried human malaria. Not anymore. These are our main problem mosquitoes in our area, and combined, there are 56 species in Harris County that are recorded, plus 1 Toxorhynchides. All right, as I already mentioned, they've been around for a long time. We've been swatting them along our evolutionary scale. And they're found just about everywhere globally. You cannot outrun them unless you want to put up an igloo in the poles areas. That's the only place where they're not found. Anybody ever visited Alaska in the summer? All right, you know what I'm talking about. Mosquitoes are everywhere. Here we have them all year long. In the northern parts of the world, they're more concentrated and come off the water synchronously all at once. Did you know this part? All males and female mosquitoes take flower nectar. They all need sugar in order to fly as adults. However, only the female takes blood. I'm sure you've heard that one before. And of course, that is that the female lays eggs, and in their larval form, there's not enough nutrients in the water for them to get the proteins that they need for vitellogenesis. Mosquitoes inhabit every water there is. We can classify them or group them based on the habitat that they're found in. Everything from permanent waters, where the Anopheles are king, or queen, I should say, salt marsh areas that the salt marsh mosquitoes and the crab-hole mosquitoes occur, the big swamp areas where there's a great diversity of really bizarre and uncommon mosquitoes that are not as worrisome as the ones that occur down here. The storm drains. We don't often think about the man-made water features as being a mosquito habitat, but for Harris County, Houston, this is our biggest problem right here. The mosquitoes that live down in our storm drains, Culex quincunca fasciatus, it hides there during droughts because what is the one thing Houstonians love than anything else? Green grass, right? So all that irrigation runoff ends down in our storm drains. The grass ends up down there. It puddles. Perfect breeding habitat. But we might have this in the neighbor may have a boat. We may have this in our own backyard. We may have a fallow yard next door. These are some of the things that breed domestic mosquitoes, not the really cool ones that are out in the forest, but the rat species, the ones that follow humanity along, That's what the problem is. We create those problems. If we could just clean up our act, we wouldn't get so darn sick all the time. Have you gotten any of those things in your backyard? I bet a lot of us here harvest rainwater in our water barrels. Let's be kind to ourselves if we do harvest water in a water barrel or water storage container, that we ensure that mosquitoes cannot reach the water level. Screen the top, plug the holes. I don't really want to go into detail of every single one. It's not about what causes mosquitoes. But you may have these because we're all plant lovers in this room, right? Tank bromeliads are a real problem. Aedes aegypti and Aedes albopictus are primary Zika, chikungunya, and dengue virus vectors. Love bromeliads. If you have bromeliads in your yard, outdoors, or near a door, please be vigilant and make sure there's not any mosquito species hiding in there or breeding in there. There's some products you can use that are non-toxic to non-targets. BTI granules, one of those. Or you can, if they're not planted, if bromeliads are not planted, dump out the water periodically. Indoors are not immune to mosquitoes. Mosquitoes will come right in that open door as you go in out. They will fly right in. If it's the right species as a container breeder, it can breed in those plant saucers. Lucky bamboo and these propagation vessels are great mosquito breeders. Even your drains. If you go on vacation, go ahead and put the plug on your drain. Ever think about your bathroom being invaded by mosquitoes? Who goes on vacation for more than 2 days? Well, geez, just about everybody, right? Be sure and close the lid. And when you come home, immediately flush your toilets, even the ones in your guest room, the ones you don't think about. Because the entry in the back right there is perfect entry for Aedes aegypti. And of course, breeding right here in the bowl itself is always a thing. All right, going back to the biology of mosquitoes. They're very acrobatic. Female, obviously, has had a blood meal. The male, look at this gymnast. Isn't he cool? Upside down. Well, he has these clasps, and I'm not sure if it's visible in this photo and all the way back. These claspers are like 2 grappling hooks. When he comes off the water as a teneral mosquito, they're turned their normal direction. As a teneral, they're just one direction. He is not sexually mature until about 18 hours after he comes off the water. Those grappling hooks turn 180 degrees. And I always go, well, but look at him. He's got to go upside down in order to mate. Why doesn't he just keep him the right direction when he comes off the water? Why does he turn him first? And of course, this is a joke, but it does happen in nature. And there's a couple of reasons for that. A, males come off the water first. There's progenitory, just like in honeybees and other flies. Males come off the water first to disperse, because incest, breeding with your siblings, is just not that cool and not advantageous, right? But also his genitalia have to turn around, and if the females come off the water and the males aren't ready to mate with them, what are the females going to do? Adios, muchacho, I'm going to go find somebody else over there. They're going to fly off, right? So the male wants to be ready and nearby, so he comes off the water first. Mosquito eggs are brilliant. They're just as diverse and absolutely stunning as the adults themselves. The biology of them is that epithelial cells that are cross-linked and to build up the chorion. The chorion is a fancy word for shell. Scientists always got to come up with fancy words, right? In the Aedes group of mosquitoes, the chorion is studded and tubercled. Culex mosquitoes or culicine mosquitoes glue eggs vertically together to build little rafts. And all the mosquito eggs, usually when they're laid, are white or clear. And then they scleriterize and melanize, become dark and black. within a couple hours. Any idea why that is? Anybody here work with fungi and lichen? Melanin is on the skin, on our skin, that helps us ward off sunlight, right? So in order to keep the egg from desiccating and be destroyed from UVA and B rays, it sclerotirizes melanin, forms, keeps the egg safe. Glycine rafts, if they're tossed out of the water, are doomed, cannot hatch. Aedes eggs desiccate, dry out, embryonation takes place after they're laid, and they remain dry until water hits them again. Here's a close-up of a glycine raft. Aedes eggs are laid singly above the water. Anaphaline's eggs are laid singly, but sport little floats. They stay on the water. Toxorhynchides, that oddball, that autogenous species, the thing that keeps me awake at night, lay white balls, little footballs that are hydrophobic, skid over the water surface and are tossed onto the water. And an even weirder group, one that's actually a problem for us in our area for virus transmission, is a subsurface dweller. The female climbs into the water through duckweed and lemma and what have you, into the water through nuphor and water lilies, holds her breath, so to speak, and adheres her eggs under the water surface, and the eggs are clustered like this. The larval structure of the mosquito larva that hatches from the egg is very similar, but it is distinct from other flies. You can still make out the head, the thoracic region here, and then the rest is the abdominal region. This is an anopheline, and here colicines, the head, thoracic area, and the abdominal section. The two differ primarily in a structural sense that anopheles have these palmate hairs, or sport these palmate hairs on these tergites, and the palmate hairs spread out and they help them with buoyancy in staying in contact with the water surface, whereas colicines sport the siphon. Anopheles do not have a siphon. A siphon is a fancy word for snorkel. I don't know why we can't just say snorkel, you know? Here's what anophelines versus colicines look like in the water column. Anopheles lack that siphon, that snorkel, so they breathe through spiracles along their abdomen. Again, the palmate hairs keep them up on the surface, whereas colicines don't need that siphon. And now I want you to think about why they do that. I'll get to that in a minute. Let's see if it works. This is a pair of anophelines larvae pulled in from a water sample somewhere. It's a local species. This is Anopheles quadripenis, goodness. And it is different from the colicines, and here's why. Did you see that? That's not the right soundtrack. I don't remember what the Exorcist soundtrack sounds like. Remember Exorcist? She turns her head 190, Linda Blair. Remember? Right. So I gave you a few seconds to think about what is the advantageous part of rotating your head like that. Well, anopheles are pollen specialists. They feed on the surface of the pollen that sheds onto the water surface. But if it always looks up, where's my next kernel of pollen? It's vulnerable to being predated upon from insectivorous fish from below. So it doesn't want to look up all the time to get its food. Hence, it can rotate its head. Here's what the larvae look like in the odd adaptation of the Mansonian Coccolithidia, the subsurface dwellers. Again, these are a real problem for us in the vector control world because we don't know they're there. We never see the larvae because they have this modified siphon that has a scalpel on the end, and it's able to attach itself to submergent vegetation in a watery habitat and attach its siphon into the cattail, per se, and extract the oxygen flow. It intercepts the oxygen there. It doesn't have to surface. So we can't kill them with monomolecular films or surface films to suffocate them. It doesn't matter. They're there when we don't see them. Mosquitoes in the larval form feed through their mouth brushes, and they create this vortex. And I don't want to go into too much detail, but both anophelines and coelescines create this vortex. But there are a couple of exceptions, and that's the autogenous species and the cannibalistic and predaceous species. They feed differently. After the larval form, the pupation takes place, and you can think of that as kind of the pupa in a caterpillar, like in a chrysalis stage. It's where metamorphosis takes place. The adult mosquito develops. But what makes them cool is that they have this really neat S-shaped air bubble that keeps them buoyant. During metamorphosis, no nutrient uptake takes place. They do not feed. But they tumble through the water through a rounded propulsion from their abdominal muscles. That's what they look like. This is Toxorhynchides. Toxorhynchides pupa are large and beautiful. And pupae no longer breathe through their siphon. Now they have these trumpets through which they penetrate the water surface. And mosquito larvae juveniles have to breathe air just like you and I. They do not have gills. So they do come to the surface with the exception of that subsurface dweller I mentioned in the previous slide. Pupae stay on the water surface, penetrate the water surface through a couple of setae or hairs on their top scutellum, push themselves up above the water when the teneral adult is about to emerge. And they literally peel themselves out like you would take a nitrile glove of your hand, that you peel your hand out of a glove. Males and females can be distinguished very rapidly and very easily. First off, if the mosquito lands on you and bites you, it is a female, right? After that, if you're looking at a flower, if you want to see who pollinates a flower and it's a mosquito, you want to look at the antennae. This is Toxorhynchides and the Passiflora incarnata, I believe. Male antennae are their noses. They got to be big. They got to smell that female. And so they're very plumose, they're very bushy. Whereas females don't really have to find the males find them. And they use other cues for finding their host, so their antennae are much more simpler. And you can see that in the picture here. This is a male Toxorhynchides rutilus. You see the bottle brush type antennae on this head. And also the palps. Female palps are much shorter than the proboscis, where male's palps are as long as the proboscis, are longer, as is the case here in Toxorhynchides rutilus. Mosquito life cycle is holometabolous. Again, I'm an entomologist. I apologize for some of the jargony word. Just means it has a complete life cycle. It has 4 distinct, separate life stages: eggs, larvae, pupae, and adult, where different things happen. We already went through that. But the larval stage in mosquito undergoes 4 distinct stages, 4 distinct molts. And in the science world, when we talk about certain ages of juveniles, we say this is L1, L2, L3, and L4 in star, or larval stage 1 through 4. But the mosquito diversity is vast. There are big ones, little ones, fat ones, skinny ones, brown jobs, usually the ones that are a problem, purple ones, blue ones, golden ones. It's a very diverse world out there, all with the name mosquito. So if you would think of the word mosquito like you would bird, you can say, what kind of bird is migrating? What kind of bird is out there? Is it a hummingbird? Is it a duck? What kind of duck? Is it a water duck? Is it a tree duck? Is it a bird of prey? You get my point. Mosquitoes are just that diverse. We just don't think about them because we're too busy killing them. But they differ not only in shape and color, but also in their vector competency. And what is that, by golly? Vector competency is this really fancy jargony word that describes this triad of things when the universe aligns, an arbovirus transmission takes place and we get sick. That's taking that mystery and defining that mystery of what just happened. We can say a vector species, a species that is a competent vector for a virus, is in play. It has to be competent for that specific virus. But it can't just randomly appear. It has to be in the right environment. It has to be the right time of the year, the right season, the right humidity, the right water condition. The rain has to be right. It's got to be in its right environment. And also, which virus or which pathogen are we talking about? Which pathogen is at play? And does it happen to infect the vector at the right time, in its right environment? And then, of course, what does it bite? If it bites the bird as a host, do we care? Probably not at the beginning, but when we talk about West Nile virus or St. Louis encephalitis, then we care again, because we may be susceptible, although we may not be the amplifying host that the virus needs to amplify, but we may be a dead-end host that can get sick. So breaking down the arbovirus transmission, and I beg your pardon if your eyes are starting to glaze over now, because we're no longer talking about biology and, flowers. But this is kind of the cool part that goes into that word mosquito. So in arbovirus transmission, let's just hypothetically say somebody's getting sick with an arbovirus, gets flu-like symptoms, gets a headache, sweating profusely. Oh my gosh, I got COVID, and goes to the doctor and says, I'm just not feeling so good. And they swab you for COVID, and that's not it. And finally they're taking blood, and they're figuring out, it's like, oh no, you went and checked your mail. You got an arbovirus. All right, so here's what actually happened. So the mosquito finds a host. It doesn't know that the host is sick or viremic. It takes the blood. The blood travels into that midgut. Remember that diagram earlier. The blood gets separated and digested, but the virus found its friendly mosquito, the one that it needs. The virus has co-evolved alongside that particular mosquito and says, ha, I'm home. I'm going to set up shop, and I'm going to reproduce inside this mosquito. It does that right there, right after, or right during which it leaves the gut wall. Because if it stays behind, it gets what? Digested. It doesn't want to do that. So it has to transcend the gut and end up in the rest of the mosquito body. It's a larger area. The virus can replicate. While it replicates inside the mosquito body, it ends up, I'm so sorry, here we go. It replicates in the body. It travels all over. It infects all the major organs, and it travels through to the salivary gland. The salivary gland is part of the feeding mechanism. The salivary gland is responsible for spitting up saliva that's full of peptides and anticoagulants that keep the blood vessels open so that the mosquito can feed. So before it is able to suck up the blood, it actually has to spit some stuff up, that saliva. That's the problem. So there's scientists out there that are trying to keep the saliva juices from flowing. There are groups of scientists out there that are looking into making the mosquitoes themselves immune to viruses, which has actually worked in Anopheles and the parasite, the protozoan parasite malaria. And there are other groups out there that just want to kill a mosquito after it's a mosquito. But, and I'm not sure what my time is. I was hoping I would have time. Anybody? Do I have time to go on or no? Sure. Okay, I'm going to see if I can wrap this up very quickly. The enemy of the enemy is my friend. Who's heard that? Everybody has. That is my motto. I'm a biocontrol agent, a biocontrol cheerleader, and a biocontrol promoter. The biological control is starting out as a crunchy granola type tree hugger. I just love all things that are alive, and taking it to the next level to see if it's actually feasible to live that way, and we know that it doesn't. So in biological control, we use nature against nature. We use the science of nature, the science of biology to help us out. Utricularia, Gibba, Utricularia radiata, and subulata occur in our area alongside 5 other utricularias, and they're carnivorous plants that occur in forested and undisturbed waters. And we can actually utilize, and BCI does this, utilize Utricularia against those subwater dwelling mosquitoes. So Utricularia has these little bladders that are booby traps, and any freshwater organism that touches those trigger hairs is actually sucked into the booby trap. And then the booby trap closes, ensnares the animal within, and starts to digest it. In the case of a mosquito larva that may not fit inside the larva, it's actually just held in place. And remember me telling you earlier that they have to breathe? Well, now they're being suffocated and slowly digested to death. This is the group of animals that is the other group of animals that I work with are nematodes, entomopathogenic nematodes, that are obligate to mosquitoes. This is Ramonomyrmex glycivoriax. It is a alien chest burster, going back to the movies analogy. The alien chest burster nematode, I'm going to have to trademark that, I think. The alien chest burster nematode, Ramonomyrmex glycivoriax, can be seen as this linky right there, and you can see the rest of that nematode actually in the rest of the mosquito larva body right there. It infiltrates the host as a free-swimming pre-parasite, bores itself into the body of the mosquito and only mosquito, feeds on all the non-essential organs, extends its lifespan a little bit longer before it's time to chest burst its way out of the mosquito host, thereby killing it. Ta-da! And that's, of course, what we want. Make these mating ball orgy things that can be like 3,000 nematodes strong. 1 milliliter, volumetrically speaking, of Ramonomyrmex glycivoriax equals about 900 nematodes. When I rear them, I go for a 1-to-1 sex ratio, equals 450 females. A single female, in 15 days only, lays 2,450 eggs. And I haven't been able to track how much longer they lay eggs. So for 15 weeks, that nematode lays eggs into the soft substrate, and a single Ramonomyrmex equals a dead mosquito. So we got 1,125,000 potential dead mosquitoes from one female eggs in 15 days. You're like, well, okay, why are the mosquitoes around then if they're there? Well, remember that habitat fragmentation thing? They're just not found everywhere, so we're putting them back. Toxorhynchides rutilus, the predator in the group, 4 larval stages are all cannibalistic. It is the group I work extensively with. I've published many papers on the rearing. There's a lot that goes into biocontrol using Toxorhynchides, and we use them in innovative releases. Some of our sites are Houston Arboretum, and Spring Creek Greenway Trail, and a few others. During their cannibalistic stage, a single larva per day can take down 30 cohorts or 30 other organisms, and they prefer other dipterans. So they feed on chironomids, midge flies, and other mosquitoes. But they're container breeders, so they're useful in use against Aedes aegypti and Aedes albopictus. They have this really bizarre overposition behavior that they're these softball players. The female never lands on the water like all the other mosquitoes do. She's a container breeder, occurs mostly in sylvan habitats, and in tree holes in the canopy. There are other creatures that might want to eat her. Tree frogs and fishing spiders also occur in those tree holes, so she wants to stay clear of being eaten herself, so she flings the egg like a softball player out of her abdomen. And she's a dang good target player. She hits the water. She can hit a tiny pinhole in a bamboo node. She's that good. And unfortunately, that the video doesn't play for y'all to see what that looks like. But hit me up some other time. I'll share that video with you if you'd like. It's so cool. We do have them in the area, and we're bringing them back. Dragonflies, as nymphs and adults are our best allies. We, in BCI, we do move them around to seed newly established water areas with the dragonflies to put them there. And the biotech, I don't think I have time to go into. It's new and emerging. It's politically charged topic. But I'm here to tell you, don't be afraid of it. If we have a Zika outbreak, if we have a yellow fever outbreak and not enough vaccines to go around, if we have a dengue outbreak in our area, which we do periodically, although it's not publicized, The technology was developed. Technology was developed at Harvard. It is safe, it is self-limiting, and it will not persist in the environment. And the modified mosquitoes cannot pass on anything to us because they cannot bite us. Here's the very last slide I'm going to show. You can help make your yard less attractive to mosquitoes and create egg sinks by putting out black containers that are attractive for mosquito egg lay for Aedes aegypti and Aedes albopictus. Just be vigilant and remove the egg contents every week. Clean them out. And if some of you will, or if you will look under your chairs, you see a sticker under your chair, possibly if you're the winner, and you will be the lucky recipient of a pair of these egg-laying mosquito traps right there. After the talk, if you will go to the book signing, you will get your mosquito traps. And with that, I would like to put up my contact information. I am looking for help. We were recently redistricted, and I lost 95% of my team. I'm back to Helen of Troy being by myself, and I just cannot do it. I want to serve Harris County constituents in a conservation aspect. I want to keep our visitors to parks safe by helping our parks and preserves reduce the biting mosquito burden, but I cannot do it by myself. I am looking for assistance in larval assays to see if some of the work we have already done is sustainable and is remaining in the field. I'm looking for some dedicated volunteers that are willing to go into the field, into the water, take mosquito samples, bring them back to me, or I will train you on how to look for the BCAs, the biological control agents, and report back to me. Thank you.